Hello and welcome to BioBiz Buzz, the podcast bringing you exclusive insights from the biotech, pharma and medtech industries. I'm your host, Jo Shorthouse, and today we're exploring one of the most exciting frontiers in oncology drug discovery, and that's the pursuit of so-called undruggable targets. For decades, transcription factors have tantalised researchers as promising therapeutic targets, yet they've remained largely inaccessible to conventional drug development approaches, but that is changing. Joining me today is Dr. Michael Bauer, CEO of Quantro Therapeutics, a Vienna-based biotech that's pioneering a revolutionary approach to targeting transcription factors in cancer. With financial backing from Boehringer Ingelheim and Evotec and a proprietary platform called QUANTROseq, Quantro has found a solution to tackling some of oncology's toughest challenges, which is the successful discovery of drug candidates targeting transcription factors. Michael, welcome to BioBiz Buzz.
Thank you, Jo. It's a pleasure to be here.
So if we can, I'd like to start with this fundamental challenge that we've just outlined. The pharma industry talks an awful lot about these so-called undruggable targets, and in particular, of course, transcription factors. Could you just talk me through why are these such important and interesting targets?
Yeah, this is a great place to start, Jo. Transcription factors are master regulators of gene expression. They basically control which genes are turned on or off in our cells. Consequently, they are of highest relevance for homeostasis, the well-regulated balance of function that are typically occurring in the cells, maintaining a healthy and functional organism. In cancer, specific proteins are mutated, or their function is otherwise dysregulated, which is driving cancer growth and metastasis. Approximately 1,600 transcription factors are currently known in the human genome, representing about 10% of all active genes. However, there are only very few FDA-approved drugs to target these transcription factors. And even worse, most of these drugs have been discovered by serendipity, not in a targeted search for drugging transcription factors. This means, while transcription factors are at the center of the problem, the dysregulation, the root cause of many diseases, there are only very few drugs available today to specifically control, modulate, or correct this aberrant function. This is both an unsolved problem and a huge opportunity to discover and develop drugs that will be able to tackle these highly relevant targets.
Okay, so this is obviously a huge medical need and a business opportunity as well, but apparently it's still largely out of reach. Can you just talk me through why that is and what exactly makes transcription factors so difficult to drug, even with all the advances in technology out there that are available today to us?
Yeah, this is a very good question, Jo. And there's still a way to go, and Quantro has found a major step advancing this field. Traditionally, drug discovery has been looking for relatively easy to find, deep binding pockets in proteins where small molecules can fit in like a key in a lock. It is fair to say that drugs for these easy targets have been discovered, and the drugs are available to deal with them. However, transcription factors frequently don't have these easy-to-drug pockets or binding site. They are flatter in their structure as they interact with each other via so-called protein-protein interactions. In this context, it's also very important to acknowledge that transcription factors are not single proteins. They are often transcription factor complexes consisting of many proteins, the core transcription factor proteins, and then co-regulatory cofactors that have a regulatory function. All these different proteins need to come together in the right conformation to eventually constitute the active transcription factor.
So this all sounds really complicated, really, really complex science. Tell me it doesn't get any worse.
It actually gets even worse. So to complicate matters, most transcription factors don't have necessarily a rigid structure. They are flexible to adapt the required shape in contact with other proteins, for example, cofactors that regulate their function. This is what scientists like to refer to as so-called intrinsically disordered proteins because they seem to lack an obvious defined structure. The reason for this is the need of the organism to regulate their function. On their own, these proteins don't have a function. They need to be flexible and remain inactive in isolation. Only when they come together, forming the transcription factors, they become active and initiate gene expression. They function kind of on demand as needed and tightly regulated within each cell. For drug discovery, this means that a potential druggable conformation may only arise in the interaction of the proteins that together constitute the transcription factor, including relevant cofactors and regulatory proteins, and all this may vary depending on the cellular context. Furthermore, the part of this process is binding to the DNA. The exact structure of the protein's DNA binding site is only finding its right conformation to recognize the target sequence on the DNA in the process of binding, when all these proteins of the transcription complex have come together. In short, only the interaction of many proteins and eventually the process of binding to DNA is forming a potentially druggable shape of a transcription factor. Now, this is extremely difficult to grasp structurally because you need to reconstruct such interactions outside of the cell. For example, if you want to crystallize these proteins for X-ray crystallography to obtain a high-resolution 3D structure. While this is relatively straightforward for singular well-structured proteins, it is much more difficult, and in many cases even impossible, for such transcription factor complexes.
You've explained that really, really well. Thank you, because I don't have a background in transcription factors. So if I follow correctly, is it still difficult, or in many cases, I guess, not even yet possible, to see how transcription factors are truly composed and structured? I mean, do we know exactly how they function? And can we measure transcription itself and the transcriptional activity of transcription factors in living cells?
Exactly, Joe. That's the point. At Quantro, we believe that nature is not intrinsically disordered. It is much more a structural flexibility with a clear purpose. It's a commonly used misconception because they simply have not been able to see what happens. So until now, transcriptional activity has not been measurable in a precise time-result fashion. In fact, scientists have been dreaming for decades of having a method that precisely measures transcriptional activity and dynamic changes of transcription at any time and with ease. It is, in a way, a very clear challenge. If you want to do drug discovery, you need a reliable assay, an assay that is truly representing what happens upon intervention with the target, an assay that clearly and without any doubt tells you what happens caused by a drug candidate hitting the target, and if it's doing its job. To give you an example, nobody would ever consider trying to develop a specific kinase inhibitor without having a suitable assay. If you want to discover a kinase inhibitor, you need a kinase assay. And the same applies to the discovery of transcription factor inhibitors or activators. To conduct transcriptomic drug discovery, you simply need a transcription assay. The problem is, until now, this didn't exist. Before Quantro developed our time-resolved transcription assay, this was simply not possible. But now, Quantro's made this dream come true. We directly measure transcription. We have developed the ultimate and universal transcription assay for whatever target we're interested in, and for whichever indication we may want to look at. We believe that the issue of non-druggability of transcription factors has largely been an issue of non-visibility of transcriptional dynamics, the absence of a reliable transcriptomic assay, and we have solved this problem.
Michael, this all sounds really, really interesting. So are you, just to be sure, are you saying that Quantro has solved the problem of making transcription quantitatively and qualitatively visible and measurable? Because that would really be amazing.
Yes, correct, Joe. This is exactly what we do. Quantro has solved the problem by taking a functional perspective on transcription. We are the only company that can directly measure transcriptional dynamics. We are the only company that can precisely quantify immediate changes in transcription, which typically occur within minutes after drug treatments. And I very much look forward to explaining this to you in more detail in our call today.
So there's one thing that I would like to better understand, because you said just now, you're the only company that can directly measure transcription dynamics and can precisely quantify immediate changes in transcription. But what about your competitors? There are many other companies that are working on transcriptomic drug discovery. They use mRNA-seq-based methods, or really well-established GFP reporter assays. Other approaches pursue structure-based design approaches focused on transcription factors. Or there are technologies like chemoproteomics, finding compounds that bind to selected targets, which include transcription factors. So how do you see those competitive approaches in the clinic?
Yeah, Joe, these are really valid questions. Let's take them one after the other. So let's start with transcriptomic discovery, RNA-seq, drug-seq, and the like. These are technically limited to the determination of mRNA abundance only, the absolute amount of RNA in a cell. However, because of the highly variable half-life of mRNA ranging from a few hours to a few days, you will need to wait at least 8 hours or longer to see any measurable change in mRNA abundance. By that time, you will not be able to differentiate primary from indirect, secondary, or even further downstream pathway effects, pathway crosstalk, and the like. This readout is simply completely blurred and too late. This method is too slow. Because of this half-life issue and the variability thereof, RNA-seq is only okay for taking a view on a steady state, and here it is limited to the determination of abundance, and no information on transcription activity can be gained. And this is not at all suitable for assessment of dynamic changes in transcriptional activity. It is simply the wrong method. It's good for other things. Therefore, we don't really consider this as a competition, as it answers a different question on a more superficial level. The same applies for reporter assays. This is about GFP protein and corresponding light signaling. It is just too slow and therefore not suitable either. Then, as you mentioned, alternative technologies like chemoproteomics, this is in fact very interesting. With chemoproteomics, you determine a binding event on a target, a molecule that binds to the target of interest. Such binding event is interesting, it may be relevant or not. We've seen in our experiments a lot of compounds binding to a target of interest without any biological relevance of this interaction. So the information of binding alone does not tell the full story. We at Quantro use chemoproteomics as a very helpful tool in our target deconvolution work. After we get the functional readout, we check on which target the drugs are binding. This means we have compounds that deliver the desired effect on the target or pathway, and then we explore the exact mechanism of action. Using chemoproteomics alone takes the opposite approach. You only look at if a compound binds to the target of interest, which may be important or mean nothing. So this, of course, would be an interesting pre-screen for us, with a focus on just one target protein and molecules that bind exactly to this target. A valid approach, but very limited. Therefore, we use chemoproteomics and other omics technologies for target deconvolution. These are all techniques that cover different features of the compound. For this reason, we don't consider this as competitors. They're relevant and complementary approaches, and we do use them as a secondary or counter screen.
Okay. Quantro has found a solution for this long-standing challenge to understand and measure transcription, but can you just explain to me exactly how it works? Could you just explain how QUANTROseq, your platform, could you explain what QUANTROseq does and what makes it different from anything else out there?
Sure, Joe. Happy to do so. So the technology was developed by our scientific co-founders, Stefan Amaris at University of Vienna and Johannes Zuber at the Research Institute for Molecular Pathology in Vienna. We take basically 3 steps on this path to Quantro's transcriptomic discovery engine. First of all, our proprietary core technology, the time-resolved direct measure of transcription dynamics. Second, the definition of search profile. How does the transcription change, and what exactly should a drug deliver, and how would it look like in the cell on the transcription level? Finally, this is translated in our assay development and screening for the identification of drug candidates that deliver the desired effect. Once we have identified these hits, they undergo the usual hit validation, medicine chemistry optimization process towards getting a development candidate and further on.
For our listeners, could you just give me a few more details, a bit more explanation into how this works, starting with your core technology?
Yeah, sure. So the core technology is basically measuring transcription. For this, we measure transcription by using a technique that is called metabolic labeling. As we work in cell cultures, these cells are getting all the nutrients supporting in their growth medium, including building blocks to make RNA. At a defined time point, this medium is replaced with a fresh culture medium containing a surrogate building block for thiouracil instead of uracil. From this moment on, the cell starts to use the surrogate building block to make new RNA, while the old RNA is still in the cell, subject to the normal mRNA turnover. If you do this for a defined time, let's say 1 hour, the cell will still have old mRNA in the cell, and of course, any newly synthesized mRNA made during this last 60 minutes. When we then stop this reaction, lyse the cells, we are using a smart trick. The 4-thiuracil in the mRNA is very sensitive to alkylation. So we conduct this chemical alkylation reaction, and then in the process of reverse transcription, making cDNA for sequencing, we get a T2C conversion. With this, we use a standard sequencing method to determine the ratio of old versus new RNA, because we see the difference on the T2C conversion on the DNA level in the sequencing. Now, this ratio we use to calculate the exact rate of formation half-life for each of the approximately 20,000 genes that are typically active in a living cell.
That really is a smart trick. I know you said it was a smart trick, but it really is by the sounds of what you're doing there. You use metabolic labeling to precisely determine transcriptional activity. But how does that then get used as an assay?
Yeah, that's a good point. So the next question is then to ask the following. If you want to find a drug that controls a specific target, you need to know how exactly the dynamics of gene transcription change if you remove this target from the cell. What is the transcriptional profile change if this target is suddenly eliminated? So as a second step, we're using a well-established technology, so-called degron engineering, for inducible target degradation. What this essentially means is that we instruct the cell to selectively degrade the target of interest in an inducible manner. To achieve that, we genetically modify the target itself by adding a small binding site for a small molecule compound called auxine that acts as a molecular glue. Of course, we do this engineering in a way that does not interfere with the natural function of the target. We have established a lot of know-how to do that. So, having established this modified cell line, we simply add auxine to the cell culture. It binds to the binding site on this target of interest, and this triggers binding of E3 ligase and ubiquitination of the target, leading to an immediate degradation of the target by the cell itself. In other words, we simply tell the cell to degrade the target, and usually this happens within 20 to 30 minutes.
This is really fascinating science that you're using here. So you actually tell the cell to degrade the target. So can you just explain to me why you do that and what is that good for?
So if you imagine the cell selectively degrades the target, nothing else happens. Auxine has no other effect on the cell. So the cell degrades the target of interest, for example, a target that's very important for cancer growth. So suddenly, as the cell eliminates this target rapidly and selectively, this cancer cell has suddenly lost a key protein that is essential for its growth and proliferation. And this, of course, has consequences in the dynamics of gene transcription. When we use our technology, we get a very clear and very clean information on the exact transcription changes that occur upon the sudden elimination of the target. In other words, we precisely measure and address the key question: what exactly happens in the cell? How does transcription profile change if an oncodriver target is suddenly eliminated? When we do this, we clearly see in the first hour which gene instantly respond and which remain unaffected. These are the so-called direct primary transcriptional response genes. And this is what we are looking for. This defines the search profile for a drug candidate that is intended to deliver exact effect in terms of changes on transcriptional dynamics. This effect that occurs when the target is suddenly knocked out. From this, we know precisely if we find a drug that delivers the same profile, it does the same, it hits the target of interest. It eliminates the target function in a way that resembles or is identical to the cell degrading the target itself. This is what we call the search fingerprint, and as this change is characteristic for each target and its respective elimination. And this information we then use for designing our assay to find drugs delivering exactly the same effect. This approach can essentially be used for any transcription factor target. It is a universal approach to find drugs for controlling the activity of transcription factors in a living cell. The good thing is, we know it works we do find compounds that deliver this effect on difficult targets which have been thus far considered difficult or impossible to drug.
This really is amazing science that you're talking me through here. I just have one more question on the science before I'd like to move on to partnerships and the more commercial side of the company. But my last question about the science is this. What do you do with the hits that you identify in the screening?
Yeah, this is not really rocket science. So once we have identified hits for our screening, these undergo the normal process of hit validation, followed by the standard optimization process in medicine chemistry, hit to lead to candidate optimization, admif profiling, pharmacology, tox profiling, etc., on the way to clinical development. So once we have identified hits from our screening, they simply undergo the normal process of hit validation, followed by the standard optimization process in medicinal chemistry, like hit to lead to candidate optimization, admif profiling, pharmacology, tox profiling, etc., on the way to clinical development. For well-known pathways and targets, this is easy to confirm and identify and validate the mechanism of action for our new drugs. For new targets, we are well equipped to engage in target deconvolution to precisely work out what exactly the mechanism of action might be. And we can use our technology in the process of compound optimization, so we can actually do this in parallel. We can optimize the compounds while still researching on the exact novel mechanism of action. And this really is the advantage of our technology. You can cover the whole range of innovation. We find better compounds for established targets. We find new mechanism of action for drugs for known targets, and we can discover new targets on known or even poorly understood signaling pathways or new targets all over. So we can serve the full spectrum from more conservative approach for just looking for better compounds on established targets, or using our technology as an innovation catalyst, opening access to currently unexploited, unknown, and new targets.
Very, very fascinating science. Thank you so much for talking me through it so I understand exactly how it works now. Let's talk about partnerships, because they really tell an important story around external validation. Boehringer Ingelheim has been collaborating with you since 2022. Could you just talk me through how that relationship has evolved and what does it mean for Quantro?
We are very proud of the partnership with Boehringer, which began in 2022 as a 3-year collaboration agreement. In June 24, we reached a major milestone. We successfully accomplished all preset goals for technical proof of concept of our platform. We completed a comprehensive cross-validation of our high-throughput screening with other methods and the identification of high-quality hits for previously undruggable transcription factor targets. And finally, we successfully conducted the first ever transcriptomic high-throughput screening actually 350,000 compounds in just a few weeks.
Oh, wow. So how did Boehringer react when they saw that?
I believe they were impressed. Professor Norbert Kraut, who was at the time global head of cancer research at Boehringer Ingelheim, described seeing this as a very convincing and robust technology proof of concept and highly attractive hits from this first screening. For us, this validation from a top-tier pharma company, of course, is tremendous. As you can see from our press release last year, we announced a 2-year extension of this collaboration. This deal is very important to us, showcasing our capabilities. And of course, financially important, as Quantro is eligible to receive up to 500 million in success-based milestone payments along the way of this project to the market.
That's 500 million euros is a really substantial amount for the company, for any biotech. And it's obviously a very good validation of your platform as well. Besides the collaboration deal with BI Oncology, Evotec, and Boehringer Ingelheim Venture Fund, I believe are your seed investors. Is that correct?
Yes, this is correct. Both companies are our main shareholders right now. They acquired significant stakes when we launched in 2020, and they've been supporting the company ever since. Evotec also provides hit identification services for our proprietary programs and joined our supervisory board, giving us access to world-class drug discovery infrastructure and advice. BI Venture Fund is our lead investor, already strongly engaged from the start, and we're now raising a 40 million Series A financing, and again with very strong support from the BI Venture Fund. They are leading this round with a 10 million investment.
Okay. So let's talk a little bit about the macro environment just for a couple of minutes if we can. The broader biotech funding environment has been incredibly tough over the last couple of years. Venture funding down nearly 45% from 2021 peaks, as we all know. How much does strategic pharma anchors change the picture for a company at your stage?
They're critical. The funding landscape has been extremely challenging in the last 3 to 4 years. Venture funding for biotech has dropped dramatically, and investors are typically concentrating on fewer, larger rounds. There seems to be a focus on few topics like well-established antibody-drug conjugates, for example. AI and machine learning has been a fashionable topic, but this also seems to cool off somehow. Many funds are actually interested in more advanced clinical stage investments where the data readout can come within a few years. Preclinical early stage, really innovative drug discovery companies are currently finding it extremely difficult to access capital, as the path to a significant value inflection point is longer and sometimes more difficult to plan. In this environment, having a validation from industry leaders like Boehringer Ingelheim and Evotec as investors and a collaboration deal with BI, this is really important as a validation and making also to the outside world visible that what we are doing is really relevant, it is successful and works and is worth to join in. With our work, we have successfully confirmed the platform works. We are addressing a real need and we can execute. We create innovative first-in-class drug candidate pipelines. Beyond the validation signal, strategic pharma partnerships also provide, of course, a source of non-dilutive funding through milestone payments, which we can use for building our own portfolio. So essentially, we're standing on two legs, pharma partnering and venture financing, and we intend to continue that model in the future. So as we are simultaneously running our Series A, we are also in discussion with a number of top pharma companies that want to use our technology to build their own portfolios beyond what's currently possible.
Let's step back for a moment and look at the bigger picture. Let's talk about innovation in the pharma industry. We have a looming patent cliff that's frequently talked about in the media and the industry events. The pharma industry is facing really enormous structural pressures. The cost of innovation, for example, pressure on pricing, and demonstrating real value for patients. So where do you see Quantro fitting into this context?
I would actually like to take the patient's perspective here. Cancer is still a leading cause of death. Many autoimmune and inflammatory diseases are without adequate treatments. An aging population with devastating neurodegenerative disorders, Alzheimer and other forms of dementia, Parkinson's disease, you name it, an endless list. And what we see is that a lot of resources, including venture capital, goes only in incremental improvements. Our industry, pharma as well as biotech, must drive innovation. And I mean true innovation, not just incremental improvements. If you look at cancer, for most of the established targets, each company has something on offer. And of course, more is in the works. This is all great, but it's more like lifecycle management, and the 7th generation of an inhibitor for a pathway that has therapeutics for 2 decades is not unimportant, but there's also other important things to be done. We need to move on. We need to really drive innovation, as there's still too many diseases without any decent treatment. So if you look around, who has the courage to engage in new target discovery? Who is working on finding the drugs that are addressing current unmet medical needs? Who is investing in companies that are set to find new drugs for new targets for the therapies of the future? We see Quantro at the forefront of this pure innovation. We open access to completely uncharted territory. At the same time, our technology can be used for innovation and improvements of known targets, of course. So we can do both. And I believe from an investor's perspective, this is very well-balanced risk and something to really pursue and go forward with. We and our investors, we can choose how high we set the bar for our vision. I personally believe that we should have the ambition to shift the bar and go for real innovation, new first-in-class therapies for currently untreatable diseases. Payers, insurers, and the patients are demanding better value for money. We need more than just little improvements. We need transformational new therapies addressing unmet medical needs And that's exactly what we are after. Transcription factor targeted therapies represent such a target space. It's a genuinely novel mechanism of action, largely untapped. We are not competing in a crowded space for incremental differentiation. We are opening access to an entirely new class of targets, which could lead to first-in-class medicines with a potential superior efficacy and reduced resistance.
Besides your activities at Quantro, it really feels like there's a broader shift happening in drug discovery at the moment. We've got new modalities like PROTACs, molecular glues, and stabilized peptides now entering the field. How does Quantro fit into that evolving landscape?
This is indeed an incredibly exciting time, Joe. The definition of druggable is expanding rapidly. What's crucial to understand is that QUANTROseq is modality agnostic. This means we can identify and evaluate small molecules, PROTACs, degraders, peptides, microcycles, network products, whatever you like, any modality that changes gene transcription, we can deal with and we can use. We can find drugs that deliver the desired functional effect, whatever the chemical starting point might be, and we're systematically building access to compound libraries across these new modalities. Where reliable 3D structures exist for a target, we also like to use rational design and AI-driven compound optimization. This flexibility is very powerful. We can identify the optimal modality for each transcription factor based on a specific structure and functional characteristics and depending on the nature of the target.
With all this in mind, and we've talked about progress within drug discovery and within partnerships and the wider biotech industry pressures, with all this in mind from our conversation today, what does success look like for Quantro in the next 3 to 5 years?
Our immediate goal is advancing our lead program towards clinical development, and we believe that a successful IND filing for our lead program is possible within the next 3 years. On a 5-year horizon, we plan on launching multiple programs running across several targets. We know our technology works, and Quantro is a go-to partner for transcription factor drug discovery. We believe QUANTROseq and the suite of assays we have built around it are the gold standard for this target class. There's nothing better, there's nothing more precise. With this, we are contributing to a paradigm shift in oncology. Each company has a specific technology angle. We take the functional transcriptomic perspective, and we are a technology leader in this field. We are starting the discovery processes with compounds that deliver the desired effect, precise, on target. This is how we deliver true innovation, new modes of action for currently undruggable targets.
That's a really bold vision. What would you say to somebody in pharma or biotech who's watching this space and wondering whether transcription factors will finally deliver on their promise this time around?
There's no doubt this is relevant, and there's no doubt it's possible. We can do this. I think there are 3 things that have fundamentally changed. First is biology. Our understanding of transcription factor networks, protein-protein interactions, and disease mechanisms has massively advanced. We can now rationally select targets based on genetic validation and mechanistic understanding. Second is the chemistry. There are new modalities, PROTACs, molecular glues, macrocycles, stabilized peptides, and all these are giving us far more tools than existed a decade ago. We are not limited to trying out finding classical binding pockets which may actually not exist. Thirdly, there's technologies available today. Platforms like QUANTROseq, combined with AI-driven drug design, cryo-EM, spatial biology, chemoproteomics, and much more. All this is giving us an unprecedented ability to screen, validate, and optimize compounds against challenging targets. Since we started Quantro, we've not only built our internal platform, we've also systematically established a network of strategic collaborations to access all these cutting-edge technologies. Just to mention one, we collaborate with the IMP on genetic screens, which is complementing our capabilities with our transcriptomic approach. Quantro is much more than a transcriptomic screening boutique. We are not a service company. We run a fully integrated drug discovery engine, building innovative first-in-class portfolios for us and for our pharma partners. The convergence of these 3 factors, biology, chemistry, and technology, this all means that we are not repeating past failures. We are operating in a fundamentally different paradigm, and new standards. And we are setting a new standard ourselves on transcriptomic drug discovery. We firmly believe, and we've shown it actually, transcription factors are not undruggable. The issue was that changes of transcriptional dynamics were simply not visible with the available methods. With our technology, transcriptional dynamics are visible and precisely measurable. The question is not if transcription factors will be successfully drugged. It is only which company will be first to do that and bring new medicines to patients.
Michael, this has been really fascinating, and I've certainly learned a lot about transcription factors, the challenges, but also what's on the horizon. Before we wrap up, what's the one thing that you'd want our listeners to remember about Quantro Therapeutics?
Well, I want them to remember that Quantro has successfully delivered a breakthrough in medical sciences. We have found a way to systematically discover new transcription factor inhibitors or activators, whatever is needed across indications. With this capability to directly see immediate drug-induced changes in a target's transcriptional dynamics, irrespective of potentially unknown mechanism of action, we are able and already are in the process of delivering both new drugs, new modes of action, addressing currently unmet medical need, targeting these undruggable targets. And we're not just developing drugs, we are redefining what is possible in oncology and other indications. For too long, transcription factors has been dismissed as undruggable, and we've changed that. We are proving that with the right technology and the right approach, these critical targets can be reached. Every patient deserves access to the most innovative therapies, and by unlocking transcription factors as a target class, we are potentially opening pathways to treatments that are currently simply not available. And this is what drives us every day.
And if people want to learn more about Quantro and follow your progress, how can they do that?
Anyone interested can reach out to me or our leadership team through the Quantro Therapeutics website. We also linked on LinkedIn, both the team as well as we have a corporate LinkedIn website for Quantro Therapeutics, where we share insights from conferences and announce major milestones. I will be attending BioEurope Spring in Lisbon in March and other partnering and investor events during the year. So if you're really interested, we're here. We're easy to reach and very open to conversation.
Fantastic. Dr. Michael Bauer, CEO of Quantro Therapeutics, thank you so much for joining us on BioBiz Buzz.
Thank you, Joe. It's been a real pleasure talking to you.
And of course, thank you to our listeners for tuning in. For more insights from biotech, pharma, and medtech leaders, subscribe to BioBiz Buzz wherever you get your podcasts. I'm Jo Shorthouse, and we'll see you next time.